Welcome to today's CELLIMA Talks webinar, Deep Phenotyping and AlphaFold. We're thrilled to have you join us for this engaging session with Marc Bickle. The session will begin with Stefan asking Marc key questions about high-content imaging and also in the context of deep phenotyping and the role of AlphaFold in early-stage pharmaceutical research. Marc will provide insights into how these advances are shaping research and the development of innovative tools in the field. Thank you for joining us, and we hope you enjoy this enlightening session.
Okay. Hi, Marc. It's a great pleasure that you have some time to share with us and to give us some insights into deep phenotyping and AlphaFold as novel research tools for in early research. I know you're an expert of high-content imaging and miniaturization, automation, automated image analysis, AI tools, so whatever, in the context of high-content screening and high-throughput phenotyping. Could you, for our viewers who might not know you as well as I do, briefly share who you are, what is your research about, and what are your interests?
Yes, hello, Stefan. Thank you very much for inviting me for this interview. I'm very much looking forward to our chat. So, yes, my name is Marc Bickle. I am currently working at Roche. I was until very recently at the Institute of Human Biology, a new institute created by Roche to specifically study advanced human systems. And prior to that, I was at the Max Planck Institute in Dresden, where I was leading a screening facility. And already back then, we were using high-content screening and started very early, actually in collaboration with you back in Bayer. That was a very long time ago. We started working on spheroids, early times on how to use hydrogel for manipulating those systems and so on and so forth. So I'm looking back on many, many years of working with diverse systems, using imaging mainly as a readout and automated liquid handling and all of the pertaining data analytics that comes with this type of activities.
Wonderful, yeah. It's been a while since we've met the first time. I think it's about 15, 17 years ago or so when we did this 3D organized project. Marc, this leads me to my first question. You're a renowned expert in high-content imaging. Can you give our viewers a quick introduction or explain some key steps that are important for using high-content imaging applications to support scientific research in early research, and to what is important so that your high-content approach is really effective and efficient?
Yes, gladly. So high-content imaging is a technology where you're applying microscopy to acquire data with your cells. So it's a cell-based screening technology where you're growing cells in high-density format ranging from 1536 plates down to 96 well plates, or also using chips in plate formats. You grow these cells in these supports, and you might treat them either with compounds or some kind of genomic tool like CRISPR, RNAi, or various mRNAs. And then generally what you do is that you're going to fix, it's rarely carried out in live cell, although that is possible too. You generally fix your cells and your systems and then do some kind of fluorescent staining either with antibodies or dyes to reveal the structure of your cells as they've grown in their support structure. And then you use automated microscopes. So these are microscopes which don't resemble at all what the general public imagines under microscopes. So first of all, these are instruments which do not have binoculars. You never look down the microscope to look at your cells. These are generally very big boxes with a door that opens, and you put in there your plate, and then it takes it in. So these instruments are able to do all of the automatic focusing, acquiring several images, maybe at different Z sides, in a fully automated manner. And they can do that non-stop, being connected to a plate handler, which when plate is read through can be exchanged with another plate. So what you obtain are very large imaging data sets. And one of the strengths of imaging and why this is an important technology in drug discovery is that images have a lot of information, hence the name high content imaging. It's the content of information that is meant under the high content, because you have not only information about the level of expression of analyte that you're looking at, but you also have the spatial distribution, in which cell type they are, in what subcellular compartment they are found. And so it just gives a very rich picture of what is going on in a cell, which then allows to have a better idea of what my compound is exactly doing in the cells and possibly avoiding bad compounds and helping to guide better the development.
And where in this whole process do these new methods, these artificial intelligence applications like neural networks, machine learning, and deep learning come into place to support these high content imaging approaches?
In the meantime, I would say machine learning has has a role in every step along the pipeline. So be it just when you're developing your assay, working on protocols, working on how you're going to, what are the concentration, what are the times, and so on and so forth. Machine learning can help design of experiments using machine learning. And then when you're doing your after the acquisition of your data, you need to segment all these objects. Segmenting means is to identify these pixels are actually part of the nucleus, and this is different from the background. So they can identify the nuclei, the organelles, where is your protein, and so on and so forth. And that step, especially in 3D, is quite challenging. And this is where AI has made a tremendous progress at segmenting efficiently these objects, and which was very difficult with classical thresholding method. So now this allows us not only to cultivate in 3D, but we can also analyze them in 3D and accurately in 3D. And this is very important. Then afterwards, when you have this data, you want to relate it to the compound. You might want to understand the mode of action. You have some tool compounds where you know the targets. You might have other type of information. So again, using machine learning, and you can then start predicting with this rich fingerprint of your image, of your cells, you can start predicting what is the mode of action of that compound, how is it working, or you can even predict maybe future compounds which you have not tested, and you think this type of structures might be useful in this asset. So I think it's all over the place on the high content imaging analysis.
Mm-hmm. And what are the latest advances in the integration of high content imaging and artificial intelligence for early research?
Oh, this is, it's a so fast evolving field. You will need to keep up. I think one of the very exciting advances which have come up is the integration of spatial sequencing, spatial transcriptomic, which is also a high content imaging technology. It's a sub-branch of that. Is being then to connect these very complex fingerprints with single cell sequencing data, which have a much deeper depth into what is transcribed in the cells, but has lost all spatial context. And there are now methods, for instance, by Aviv Regev 2 years ago, I believe, Tangram, which allows to integrate these data sets so that you can now project the likely expression pattern on the single cell level based on single cell sequencing data and some form of spatial transcriptomic and various forms of spatial transcriptomic data. And so this allows to suddenly have, because the data, so this allows us to have a much deeper view into the cells, which is maybe one of the weaknesses of imaging, is that you have the spectral overlap. So you cannot acquire too many channels. Typically, you will have 4 to 5 channels. You can do iterative cycles to try to increase the number of channels. This is how you then get to spatial transcriptomic data sets, and there are different technologies out there. But it's not going to go genome-wide, or it's very challenging to go genome-wide. And this is the great thing about this, the joining of these two technologies. This allows now to have a genome-wide view on the expression patterns in cell, in space.
Yeah. It's interesting, and found it funny. I had a discussion with a customer of mine who asked me how many colors can he use in such a high content imaging approach, because he has got an offer about 24 different colors that are doable, and I said stick to 3 to 4, because otherwise you will end in a mess of fluorescence, channel talk, intercrossing, and whatever. Also the different antibodies and dyes you have to use and you have to put into your cell.
So there maybe I would have agreed with you a few years ago.
Okay.
Now I disagree. And I know a lot of vendors have put a lot of effort because they know multiplexing is important. They know that these stripping protocols are very challenging. Some of them are really technically very challenging to acquire. And they're trying to do it with a single round of stainings. And they come with linear mixing and technologies, and again, using machine learning behind it. They are achieving on a routine, something like 7 to 8 colors on certain type of instruments, certain type of lasers and filters. So also in this respect, there have been progress. And this is already a very important progress, in my opinion, because it allows you, some people will say, what's the difference between 4 and 8? But in my opinion, I know I've been very often, I would have loved to have 8 channels information. It does enrich a lot, especially if you choose well your markers that are nicely orthogonal. You will learn a lot more about your phenotype.
Yeah.
It won't allow you to fuse with a single cell sequencing data set, that is for sure, but it is more scalable in the number of compounds you can use.
Yeah. I agree with you. If you're an experienced researcher in the field of high content imaging, or let's say with fluorescence dyes and antibodies, and you have the equipment and the sophisticated tools in place, then it's doable up to 6 to 7, 8, you said. But 24, I think, currently, I would hesitate to do for that. And if someone wants to start in high content imaging, I would stick to 3 to 4 at the max for the first.
Yes. I mean, already with 3 to 4, there is, for instance, a paper by Sims et al. Where they have a 3-color high-content imaging data set where I've done the deep phenotyping. Because also with 3 colors, you will find a lot of publications who still have a single parameter, even though they would have at their disposal many, many more parameters. Well, using just this 3-color, but a very deep phenotyping, as extensive information as possible, and using historical data on in vitro data from some of these compounds on some of the targets, they were able to predict hits. And in 2 ongoing screening campaigns, they increased the hit rate once by 60-fold and the other time by 250-fold. Mm-hmm. So you reduce a deck of 1 million compounds, right? You are suddenly dealing now with something which is in the range of 40,000 compounds, or even less in the case of 250. So we're becoming really in a realm where we can start again having more, maybe deeper phenotyping, but already 3 gives a lot of information.
Yeah, it brings me back to our red thread of our interview. What is the impact that high content imaging and artificial intelligence tools have on the accuracy and the interpretation of data that are coming from these approaches?
Well, there is, if you look at, I'm going to pick out, for instance, from Fabian Theis, for example, the CAMPAS framework, which identifies consistent landmarks, cellular landmarks. Because when you're doing sort of a deep phenotyping approach where on each pixel you have a row of 80 different channels, and each pixel is and these things, it becomes very difficult to analyze that. And these are methods which were developed to realize, okay, this actually is something which I find a lot, but sometimes that channel is missing, sometimes that channel is brighter, sometimes that channel is thin, or it's a bit, and thereby it actually tries to ignore the variability due to the compounds to find things which are consistent across it. And then they look at these things which are consistent across it and ask, how is it different in my control versus the compounds? And these are consistent things which end up being organelles and stuff like that, which are so that emerges from the data. So that's one thing where I think it helps you to interpret the data. You find that it is being by AI determined structures which you can wrap your head around. So it's not a black box. You know what it was, what kind of because you have the markers. You know what it is representing, whether it's a Golgi, whether it's a nuclear organelle or something like that. And then the other example again from the Theis lab is where they're predicting cellular responses based again on data that you have acquired, where you're doing where you're extrapolating from the data you have, and then predicting these are targets. These are going to be the ones, the pathways which are going to affect this biology. So again, then you have now suddenly you're kind of fusing the target drug discovery world with the phenotypic drug discovery world, because you're using phenotypic drug discovery to predict molecular targets, which then you can then try with the molecular target. Maybe we might know structure, or we might predict the structure using, again, AI tools. And then we might, again, use AI tools to predict compounds and structures which would bind, inhibit, and change these things. So you reduce both the size of your screening deck, you get more certainty into what might be your target. This is always one of the big drawbacks of phenotypic drug discovery. You have to go and find your target, or at least try to really understand what the drug is doing. And you might have actually a convergence of these two fields by using AI tools. This is very exciting times, I find.
Have you methods in mind with which you can efficiently reduce the data when analyzing these complex systems without affecting the results?
I mean, in terms of data reduction, an image can be compressed, and there are companies out there and who have algorithms, and again, using AI for compressing the data. And depending on the type of things you're dealing with, you can have a compression of 10%, down to 10%. So if you're dealing with petabyte size, that costs a lot of money to maintain, to keep live, and so on and so forth. If you can reduce that already to a few hundred terabytes, it's really a big gain. So that is certainly also one thing where you can. But I think what it does is, I think what you need to do is to make the effort to create a foundational data set. It's a bit what the JUMP consortium has done with the cell painting assay. You create it with a diverse chemical space, you do a deep phenotyping approach. Now you can start mining that and trying to extract other things. So I think these kind of approaches, where you're doing a deep phenotyping, and we'll have to go through the deck with maybe not 20, but maybe 3, 4, 5, let's see, colors. But then all the following screens will then be much smaller because you're only using a subset and you're much more informed. You have modeled you basically have modeled your chemical space through the biological space. So now you can fit one to the other, and then so it's more, you need to have an upfront investment, which will pay in the long run, because then you don't need to have such large data sets.
What's your opinion? Just some speculation, some ideas. How does AI-based image analysis can affect data reduction and interpretation in the context of early research? I know this is a question that can be hardly answered right now, but just some thoughts about that. Can you share this with us?
I don't think AI is primarily about data reduction. It actually, the reputation is just the reverse. You need to have a lot of data to be able to apply and leverage these technologies. So I don't think this is where the advantage is. I think it's more about the likely accuracy of the results and the interpretations that you can extract from the data. I believe are because this latent space really, if you add the information, will capture a lot of the biology that we know of, and so will help you to have more informed decisions, I would believe.
This leads me to the headliner of our interview, phenotyping. What is meant by the term deep phenotyping?
Well, phenotyping is looking at a shape of something, and phenotyping, from formation in my early, I was a geneticist, but that's what geneticists do, right? We compare. I mean, I worked with yeast. You don't have much to compare with a yeast colony, but C. elegans, you have phenotypes. One might be uncoordinated, one might be stumpy and elongated, and so on and so forth. This is a phenotype. Now, deep phenotyping is that you just describe with as much accuracy the shape and the look of the object you're looking at. So you're looking at, and you're using mathematical descriptors of shape, how, the axis of ellipse that you would fit around the objects, what is the major and the minor axes. You look, of course, at the intensities. You look at the spatial relationships between how far away are they clustered? Is there some kind of pattern that can be recognized in the distribution of this object? The intensities, and so that adds up to every parameter is a number, and you add up to vectors which are thousands of parameters long. And that is what is meant in deep phenotyping: is that you have exhaustive description. And you can have, as we discussed at the beginning, exhaustive description on very few readouts. But you can imagine we were talking about AI. AI is all about tensor mathematics. This is also a tensor. Now you have more and more channels with more and more things, so you just get a gigantic object, which is basically mathematically intractable. So that's why you have to use AI, the curse of dimensionality. So this is where you have to apply AI, where patterns are learned out of it. So you're not using the entire vector, but you're using the patterns which are inherent, and you learn those patterns in the data to then come to your conclusions.
What main challenges do you see in implementing deep phenotyping into early research processes? And in your opinion, how can they be overcome?
So I think, again, AI plays a role. So I'm just thinking of recent publication by Loïc Royer on integrating large language models in image analysis. So it's a specially trained Omega, I believe it's called, version of Napari, where a large language model has been trained specifically on image analysis using Python. And you can just interact with that entity, and it will create you solutions for, you describe your problem. I have nuclei here, and I would like to have them segmented, and would like to have all kind of measurements possible on intensity, shape, and so on and so forth. And actually, to be able to play with the data, I would like to have a widget where I have a big radio buttons to play around with my thresholds or whatever kind of thing. So this is sort of democratization. There is a democratization ongoing where more people can access more complex technology because they are AI assisted. This is what we see in the consumer world, where people can do actually quite amazing things without even realizing on their phone because they're AI assisted. We see it now in emails being very well drafted, AI assisted. So it is very challenging. We're very early days. All of this publication we were talking about, the oldest is now maybe 6 years old. So it's very new. It's a lot of things are being tested. A lot of going to be some errors done, and oh no, let's do it this way rather than this way. But I think it should end up in something where the more people will have access to these technologies. This is really the hope. And this is something I was also really striving towards also in my work. There's always to use the most user-friendly tools, offer courses on how to use them, and try to hide a lot of the complexity that the user doesn't necessarily need, especially when just executing an assay where you just need to tweak a few parameters. Developing it is something different, I agree. But so I think it will be possible. In terms of hardware, it is, you use the normal hardware. There hasn't been anything specially invented for doing this. Well, everybody has access. It's about protocols, very good maintenance of your instruments. I think it's when you have now CROs offering also these services and so on and so forth. I think it is going to be more accessible to people if it's because it's just too it's just too valuable.
Mm-hmm.
But there's always a risk with any technologies that it stays, and it's of course at the moment is the richer places with institutes which can afford this kind of technology, but there's mainly the ones who are developing it. Once it's developed, it then can move because there's a lot of sharing in the community. It can move out into other labs where we don't have the resources for doing the research on it to develop the technology, but actually applying it might be possible, especially in central facilities and such.
Yeah. Have there already been successful cases published where new therapies have been identified by this deep phenotyping approach?
It's probably too early days, but I am certainly not an expert on what right now has been approved by the FDA and what role did AI play in the development. I would imagine it's such a recent development, AI, and it takes such a long time to get a drug approved. It's probably too early to say. Some of the ones which have been recently approved may be in the later stages. AI was brought into analysis of clinical trials, but generally you have to declare these things way in advance. So I would suspect that it's too early yet to have, but I'd be happy to be wrong.
Yeah, give it the next 5 years or so. Yeah. Marc, coming to the next big headline now of our interview, what is meant by AlphaFold? It's a big word, I guess.
AlphaFold, yes. So AlphaFold was again AI that was published in 2018 for structure prediction. And already the very first version was made a big splash because it was very good at predicting structures. Then came in 22 AlphaFold 2, which was even much better. That was a big game changer. People in the structural world, when you read a bit of the comments, they were really very impressed by the performance of this system. And now just in May this year was published AlphaFold 3. And AlphaFold 3 builds upon the AlphaFold 2 and expands it and also modifies many aspects of AlphaFold 2 to have a system which can now also predict complexes and complex with various biological molecules, be it with nucleic acids, with other proteins, or with compounds. So, and this is just very recent, but I'm sure this is going to have a gigantic impact, because if we see this deep phenotyping with this now predicting targets, and now we can use AlphaFold 3 to start predicting ligands to that target. This is what I meant by doing these conversions of phenotypic drug discovery, target drug discovery. We can now do target drug discovery inside the cells using phenotypic readouts. I mean, that's a promise. We'll have to see. It's all brand new. But this is where I find it very exciting, because that would really, really greatly reduce the number of screens that needs to be done for a compound. Whether these are going to be performing better in the clinic, Still depends on how good was your model, how good are your assumptions, and then so on and so forth. So it will still be failure. But I think one of the because you can get much faster to something that where you might be tempted to go into the clinic and reduce massively those costs. You might be tempted to go so fast in clinical trials, but this is not my topic. So I don't know all of the hurdles then to take maybe many more compounds which are coming from the early discovery into the clinic. I think no CEO of a pharma would complain if they're flooded by early discovery compounds of high quality wanting to enter the clinic.
Yeah. And is this the way how these AI tools, particularly AlphaFold and phenotyping, can improve the prediction to develop new therapies?
It's the hope. I mean, it's one of the things, of course, where I also, again, for patients, what it's doing is making earlier diagnosis. So we read a lot of cases where there are publications, of course, but where there is a hope that we can start predicting also by phenotyping liquid samples from people, urine, blood, and can start very early diagnosis of some diseases, which would then be again amazing because the earlier you catch them, the more you have chances of winning and beating the disease.
Mm-hmm. From a general perspective, what advances with AlphaFold and beside AlphaFold in the context of AI-based tools, which advances have been made in structure prediction, and how have these advances affect the efficiency in screening?
So I'm not so familiar with tools around the structure prediction world, right? Mm-hmm. I mean, AlphaFold, of course, was a big thing, so I was reading into it, but I wouldn't be able to comment too much on the other advances. And I know there are all the time publication coming out with further predictions and so on and so forth. I think it's mainly the important thing is advances on the docking, because we have been using physics-inspired methods for doing simulations for docking compounds onto molecules and so on and so forth with some success. Yeah. And now these tools are entering that arena, and from what I'm reading a bit, they seem to be outperforming in some cases sort of the more physics-informed tools that have been used actually.
Okay. So let's share some thoughts about the future perspectives of these AI tools, high content imaging and so forth. What, in your opinion, would be the next major developments in high content imaging, artificial intelligence, you could anticipate that would further enhance the efficiency and the accuracy of such approaches in early research?
I think, we were talking about the challenges of obtaining these data sets. This is one thing which is quite challenging. And I believe their advances in this field would help that, because you could then more easily get multiplex data. You could feed the beast more easily. That's one thing. I'm sure there's a lot of things that can be done on the networks themselves. It's talking to mathematicians. They find them that they are not sparse enough, and that the random initializations of weight should be actually simulated to not have a random initialization and things like that. There are very interesting advances also in physical objects being used for machine learning, where you have chaotic acoustic cages or chaotic light cages, which can learn patterns and then predict new patterns. So it's wild what's going on at the moment, so there's going to be a lot of advances also into types of networks we're using, and the large language model is one of the latest big development, and its roles in different applications still being investigated actively. There was one that just came out, I believe, last week, where a large language model was used to predict and ameliorate antibodies therapeutic antibodies talking to that was SARS-CoV-2 protein, and predicting better how to have neutralizing antibodies in the light of problematic variants of concern. I believe there is just going to be a lot of advances there. One thing which is, how good are they at predicting the mode of action? How good are they at really understanding what is going on? I believe that is going to be interesting, because I see still a big problem that we have in our high content imaging data sets is that we're missing two classes of very, very important components in the cells. That's on the one side, the metabolome, and on the other side, the lipidome. These are very important compartments to which we're completely blind in most of the cases in our screening assays, and which we know from the biology have a gigantic impact. And we ignore them because we basically don't have the reagents to detect them. And that's something which I believe we'll see how important, whether you can infer the lipidome and metabolome from the rest of the data that you have, the transcriptome and the proteome. But we're missing half of the most important molecules classes. Yeah. Now, I think this is one of the problems we are having in having more accurate predictions in preclinical data.
Yeah, interesting. Yeah. Marc, how do you perceive the role of artificial intelligence within the context of personalized medicine or in the development of tailored therapies for individual patients?
I think it will play a role. Again, it's this capacity of leveraging this complex real-world data that patients represent, so that you have a transcriptome of a patient, for instance. Yeah. The same transcriptome in two different patients can have completely different outcomes if you don't know what else is also the life, the sex, the age, the occupancy, and so on and so forth. And there is where AI is very good. I was also reading papers in, for instance, diagnosing exactly sort of neurodegenerative diseases, whereby just filming eye movements or analyzing videos of eye movements or hand movements doing a certain task, they were extremely good at predicting that Alzheimer's is occurring in this person, where they had a zero clinical thing. And in long-term trials, indeed, these people did go on and develop the disease. And basically, the AI was predicting that years in advance, and we know what makes a difference for what kind of disease. So, and with everybody having an iPhone and, or let's say not just a phone, everybody having a smartphone. This was not an endorsement. With everybody having a smartphone, you have something at the tip of your fingers. And again, you see publications of smartphone therapeutic or medical applications, which can also be a problem because a lot of the health applications of it. But if it's a regulated environment for things where the medical decision can be taken, the telemedicine is a big word, a big buzzword, where try not to get the patient into the hospital if it's not necessary. Try to diagnose at home, try to make as much as possible by leveraging these technologies and using that as confidence. Obviously, in pathology, digital pathology, AI plays a gigantic role. Also more accurate analysis of pathological samples.
Yeah. The earlier you are in the process of the onset of a disease and you can interfere with the disease, the more is the probability of success that you can cure the disease. And this is where I'm also convinced that AI is very helpful. And hopefully we will see more in this in the future. Marc, thank you so much for this very intensive interview, sharing your thoughts about the topic of deep phenotyping and AlphaFold and sharing some time with us. Thanks so much.
Thank you very much. It was a pleasure.
Thank you all for joining us today for this CELLIMA Talks session on deep phenotyping and AlphaFold. We hope you found the insights shared by Marc valuable and inspiring for your own research and work. If you have any further questions or would like to connect with us, please feel free to contact us after the session. Thank you again for joining us. We look forward to seeing you at future CELLIMA Talk sessions. Have a great day and goodbye.